Okay, so good morning, everyone. So we'll continue with the discussion for Clostridium species. So we have finished discussing the Clostridium tetani and Clostridium botulinum. So now we'll proceed with your Clostridium perfringens. So Clostridium perfringens is also known as gas gangrene bacillus. So these are short, plump, and encapsulated na non-motile organism. So, di ba, siya tong exemption sa general rules of genus Clostridium. So sila all are non-encapsulated except for Clostridium perfringens and all are motile except for Clostridium perfringens. So, siya yung non-motile. And then, it is not able to produce spores in ordinary media because normally, um, organisms can produce spores in vitro. However, your Clostridium perfringens cannot produce spores in vitro. So, the formation of spores by your Clostridium perfringens um, is during in vivo or sa loob ng human host. Ayan, and this is the most commonly isolated Clostridium species, no, under your anaerobic organisms. Ayan, so the disease that this causes is your myonecrosis. Ayan, so myonecrosis, or what we call as the gas gangrene. So this is characterized by severe muscle destruction and production of gas pockets in tissues. And your Clostridium perfringens also can also cause food poisoning and post-abortion sepsis, intra-abdominal and pleuropulmonary infections, and your colitis. Ayan. So these are the following diseases that Clostridium perfringens causes. So, virulence factors of our Clostridium perfringens is the production of alpha toxin. So, the alpha toxin that this organism produces is all produced by the 5 strains of your Clostridium perfringens. So, we have 5 strains of your Clostridium perfringens. We have your type A, B, C, and D. So, D and E rather. So, those 5 strains of your Clostridium perfringens causes and produces your alpha toxin. And aside from that, no, it is also able to produce your alpha or your lecithinase enzyme. So this lecithinase enzyme, this will split the lecithin into phosphorylcholine and diglyceride. So remember that lecithin um, is a major component or constituent of the cell membrane. Thus, having this enzyme will dissolve lecithin in your cell membrane, thus will lead to cell destruction. So aside from that lecithinase, no, it is also able to produce hemolysin, which will cause now the hemolysis, no, or is responsible for the hemolysis of the blood in the blood agar plates, cardiotoxin, collagenase, enterotoxin, and proteolytic enzymes, fibrinolysin, DNase, and RNase. So this enterotoxin will cause now the diarrhea of your infections with Clostridium perfringens. Okay, and then, yeah, so these are the following virulence factors of your Clostridium perfringens. Yan, so again, these are the types or strains of your Clostridium perfringens. So we have alpha, these are produced by all of the strains. Then mag-differ na lang sila with the different toxins that they produce. So your type A is produces alpha and theta toxins. Type B, alpha, beta, and epsilon toxins. Type C, alpha and beta. Type D, alpha and epsilon. And type E is alpha and iota. So the alpha toxin again is a necrotizing toxin produced by all 5 strains. While your beta toxin naman, which is produced by your type B and C, is a lethal necrotizing toxin. Theta toxin produced by your alpha toxin is an oxygen labile cytolysine. So this toxin can damage blood vessels, resulting to leukostasis, thrombosis, and increased perfusion and tissue hypoxia. Theta toxin can also stimulate cytokine release and causes shock. And aside from that, epsilon toxin, this toxin is a pore-forming protein and causes potassium and fluid leakage from the cells. So it causes imbalances in our body that will lead to leakage of body fluids and loss of potassium in our patients. And yeah, so these are again the following toxins that are caused by the different Clostridium species. So clinical infections. So we have again wound and soft tissue infection caused by the alpha toxins, food poisoning caused by type A strains with spores and minimal amount of theta toxin. And necrotizing enteritis or pig bell. Ayan. So ingestion of inadequately cooked pork. This is usually acquired no, through pig feasting. Ayan. So signs and symptoms are due to the beta toxin of the type C strain of your Clostridium perfringens. Then lab diagnosis of our organism, no, Clostridium perfringens. So specimen that we use for the identification of this organism includes wound, tissue, and pus aspirates. So since this is anaerobic, kailangan aspirates yung gamit natin. Swabs are contraindicated for the diagnosis because again, it will be exposed now to oxygen. So in gram staining, we can see large gram-positive bacilli and cultural characteristics. So in your anaerobic back, they will produce circular smooth colonies. And unique to Clostridium perfringens is that it causes target or double zone of hemolysis. So ang double zone hemolysis sa imuhang organism, we have alpha and beta. So it duha ka ang hemolytic pattern. So the inner zone of the hemolysis of your Clostridium perfringens is beta hemolysis. So there will be complete hemolysis. And then the outer hemolytic pattern is alpha. Yan. So the inner is beta and then the outer is alpha. So beta hemolysis is caused by your beta hemolysin and the theta toxin produced by your Clostridium perfringens. And then the alpha hemolysis naman is caused by your alpha toxin and lecithinase production. And in your chopped meat glucose agar, so it produces abundant growth and it is positive with gas production. And yeah. So next we have other lab diagnosis. No, they are or your Clostridium perfringens is saccharolytic. So they can ferment glucose, lactose, maltose, and fructose. And in egg yolk agar, again, it demonstrates lecithinase activity. So lecithinase activity is characterized by opaque yellow halo on the egg yolk agar. And your Clostridium perfringens is lipase negative organisms. And again, they are non-motil. So biochemical, again, they can ferment glucose, lactose, maltose, and fructose. In milk media, they can produce stormy fermentation. So positive result of this um, testing no, is a tear, or crack in the curd, usually seen in organisms Clostridium perfringens. Okay, and then lecithinase test again. So this is the lecithinase. It will produce again opaque yellow halo surrounding the colony on your egg yolk agar. So next is Nagler reaction or Nagler reaction. So Nagler reaction is a biochemical test used to identify organisms which liberate phospholipases or the lecithinases. Examples are your Clostridium perfringens. So the principle of this test is that the alpha toxin of your Clostridium perfringens has phospholipase activity and hence will help in the differentiation of Clostridium perfringens from other Clostridium species that produces lecithinase. So the production of phospholipase activity can be neutralized now by the activity in the presence of the antitoxin. So lecithinase activity can be done through a Nagler plate. So this is a Nagler plate. So Nagler plate can we have to strict no. We will divide this into two. So half of it will be strict with antitoxin produced by your type A Clostridium perfringens, and half is the suspected culture. So those organisms that is able to neutralize your antitoxin will not grow on the Nagler plate. However, no, though your Clostridium perfringens can produce lecithinase, it can also produce phospholipase. So the phospholipase can also um, um, remove the antitoxin present in this agar plate. That's why there will still be growth or precipitation in this area. Yeah, so the other half that has antitoxin will be neutralized. However, again, in this area, there will be precipitation still because of the presence of a phospholipase. Okay, so ayan. So this one is we have sporogenous, we have a negative reaction to the Nagler plate. So again, the positive reaction of your Nagler plate or Nagler reaction is precipitation on the plate without the antitoxin. Yeah. So next diagnostic feature or diagnostic test for your Clostridium perfringens is the reverse camp test. So nadungog na nato si camp test for your Streptococcus agalactiae and your Listeria monocytogenes. However, this is called reverse camp test because the known organism used for this test is your Streptococcus agalactiae or your group B strep. Diba originally, ang CAMP test is used to identify your Streptococcus agalactiae. However, in this case, baliktad. Okay? Si group B strep ang nagiging known organism. That's why gitawag siya og reverse CAMP test. Okay? And then, there will be arrowhead hemolysis now seen in the growth between the Clostridium perfringens and your group B Streptococcus. So what is the test principle again? So the CAMP positive group B strep is the known organism. So it is strict at the center of the sheep's blood agar, and then perpendicular to it will be the unknown organism or the possible Clostridium perfringens. So after 24 hours of incubation, should it should have a bow tie zone of enhanced hemolysis pointing towards Streptococcus agalactiae. So this one, this one. So like a bow tie na enhanced hemolytic pattern in your Clostridium perfringens. So this is because of the alpha toxin produced by your Clostridium perfringens. Okay, and then it will interact with the CAMP factor produced now by your group B strep. And they will produce synergistic hemolytic pattern. Yan. So this is the positive result of your reverse CAMP test. Okay, so that's for your Clostridium perfringens. So now we'll proceed with your Clostridium difficile, the last clinically significant Clostridium. So your Clostridium difficile is first isolated in 1975. This is usually associated in antibiotic associated pseudomembranous colitis. So this will cause colon perforations that will lead to sepsis and death. Yan, so they are motile, so with peritricus flagella and is non-encapsulated. So the toxins that they produce is the toxin A and B. So toxin A is a potent naman na enterotoxin. So this is the main cause of the colitis no, caused by your Clostridium difficile. However, it is weakly cytotoxic. So the presence naman of your toxin B can help no in the weak na cytotoxin activity of your difficile. So, your toxin B naman is responsible for the cytotoxin activity of our Clostridium difficile. So, your toxin B is actually the one that is used not for the diagnosis of your Clostridium difficile infection. So we are detecting the toxin B production of our organism. So clinical infections caused by Clostridium difficile is again the antibiotic-associated pseudomembranous enterocolitis. So the use of ampicillin, clindamycin, and fluoroquinolones no, can lead to the disruption of the normal flora. So again, normal flora is present diba, in the gut. So the gantag bacteria that is normally residing in our colon. However, because of the use of antibiotics, no, ampicillin, clindamycin, and fluoroquinolones can lead to the removal or killing of those normal flora. So your Clostridium difficile, once ingested, no, um, it will be dominate or it will dominate now our colon, causing now pseudomembranous ulcerative colitis. Yeah. So instead of the normal flora protecting us from Clostridium difficile, since naatay mga giinom na antibiotics, so ma-remove or mawala tong normal flora leading to the overgrowth now of your Clostridium difficile. So that's why this is associated with antibiotics. So this is not really induced no merely by the presence of Clostridium difficile because normally we have normal flora but the pag nawala na ang normal flora sa atong gut, there will be now overgrowth of your Clostridium difficile. Thus, it will cause now your pseudomembranous abdominal or ulcerative colitis. So lab diagnosis of your Clostridium difficile includes the following. So specimen will include liquid or semi-solid fecal specimens. So stool specimens ang ating ginagamit for this one. So microscopy, we have gram stain, of course, gram-positive bacilli under the microscope. So culture, we can use your cycloserine, cefoxitin, fructose agar. So CCFA agar. In CCFA agar, they will produce yellow colonies and it has a horse manure odor. Ayan. And then in CDSA, Clostridium difficile, agar, so they will appear, the colonies will appear rose to yellow colonies. And then serology, again, we can use a demonstration of the toxin, rapid latex agglutination test, and toxin A detection. Toxin B detection. Okay, and then cell culture, gold standard for the toxin identification. Okay, so it will lead to high sensitivity and high specificity of our diagnosis for your Clostridium difficile. So other biochemical tests, ayan, so we have gelatinase positive, lecitinase negative. So compare with your Clostridium perfringens, it is lecitinase positive. Lipase neg, indole neg, and it can also ferment your glucose and fructose, but it cannot ferment your lactose, maltose, and xylose. So spores are oval and subterminal in position, and then they are motile again, and they fluoresce under UV light. So under UV light, they produce a chartreuse, chartreuse color under UV light. Okay, chartreuse color under UV light. So treatment no for your infections with Clostridium difficile is the discontinuation of the administration of the offending drug. And we can use metronidazole for the for the Clostridium difficile no directly against your Clostridium difficile organisms. And gastrointestinal stimulant, we can also use stool softeners and enemas. Yan. So these are the following treatment no, for the infections caused by your Clostridium difficile. So you can just treat no, in the miscellaneous Clostridia on, I think this is Jowet's, or you can just use additional reading materials in your Mahon and Delos. Okay, so now we'll proceed with the anaerobic gram-negative. So, since we are talking about anaerobic or anaerobes na, we'll also discuss now the anaerobe na gram-negative naman na bacilli. So, previously, kato si Clostridium, they are gram-positive na anaerobic. And napod tayong mga anaerobic na non-spore forming katong mga organisms that we have discussed, no, depending on the metabolic product they produce. So yung Propionibacterium, yung Bifidobacterium, those are the following non-performing na anaerobic bacilli. So now we'll proceed with the discussion of anaerobic gram-negative bacilli. So your anaerobic gram-negative bacilli is a major normal flora in the colon and oral cavity. However, infections may develop, no, following surgery, trauma, or disease. Once they get out of their um, normal habitat, yan, so they will cause infection. So they are identified using colonial morphology, gram stain appearance, pigment production, antibiotic susceptibility, hemolysis, and biochemical testing. And gas liquid chromatography may also be used for the identification of these organisms. So under your anaerobic gram-negative bacilli are the following genus. No, we have your Bacteroides, Fusobacterium, Prevotella, and your Propyromonas. So we have the Bacteroides fragilis group. So under your Bacteroides fragilis group, actually daghan siya. But the most common isolate is the Bacteroides fragilis. Yan. So this organism is extremely virulent. No, once it causes infection, this will cause widespread tissue necrosis. So virulence factors of your Bacteroides. So we have Fimbriae and agglutinins, which will help adhere, no, to the host cells. Capsule, the presence of capsule can also help, no, in the um parang i-prevent niya ang phagocytosis. So hyaluronidase, enzymes hyaluronidase, chondroitin sulfatase, and endotoxin lipopolysaccharide, since it is a gram-negative organism. So the release of the endotoxin is associated with the diarrhea that the Bacteroides can cause. So it can also produce beta-lactamase, no? So it is resistant now to penicillin. Ayan. So clinical infections, again, it will cause abdominal and urogenital abscesses, peritonitis, appendicitis, and tissue abscesses. So lab diagnosis, again, in the gram stain, they will appear as gram-negative rods with rounded ends and may be pleomorphic with vacuoles, no, under the microscope. In your kanamycin, vancomycin lake blood agar, they will appear as large, wet, and gray colonies, and they are usually non-hemolytic. Okay, your KVLB is kanamycin, vancomycin lake blood agar. So in your potency disc test, no, they are resistant to everything. So resistant sila to kanamycin, colistin, and vancomycin. So in BBE naman, Bacteroides, bile esculin agar, they are bile tolerant. So they are able to grow on bile. Normally your anaerobes uh, can be inhibited, no, by the presence of bile. But your Bacteroides is bile tolerant. So, okay, they can grow in the presence of 20% bile, and Bacteroides bile esculin agar, they can develop black colonies with black halo. So again, ilang biochemical reaction, so they can grow in 20% bile. So bile esculin hydrolysis positive sila, catalase positive and negative sila with these enzymes. And negative din siya to indole, and there is no pigment or spore production of the organism. So next is your Fusobacterium. So Fusobacterium species are found as normal flora of the upper respiratory, gastrointestinal, and urinary tracts. So we have I do have a lot of species now, but these two are the most noted na isolates of your Fusobacterium. So we have Nucleatum and Necrophorum. But the most common one is your Fusobacterium nucleatum. Yan, so your Fusobacterium nucleatum is an important pathogen in biofilms in periodontal diseases, root canal infections, and dental abscesses. And this will cause also no aspiration pneumonia. So this is the causative of agent of your aspiration pneumonia. So Fusobacterium necrophorum naman, ayan, so it will cause septicemia, metastatic infections such as lung and liver abscesses, arthritis, and osteomyelitis. And it plays a role in Lemire syndrome. So Lemire syndrome is a rare but very tawagan na, very um, infective no infection of the throat. Ayan. So, in the presence also of a commensal spirochetes, your Fusobacterium necrophorum can may cause Vincent's angina or trench mouth. So, Lemire syndrome, ayan. So, healthy throat is ganito, but this one, inflamed tonsils or sore throat, this is caused by your Fusobacterium necrophorum or the Lemire syndrome. Yan. And your trench throat or the Vincent's angina naman is this one. Ayan. So there will be ulcerations in the throat. So in the presence of commensal spirochetes, no, dalawa silang mag-cause ng Vincent's angina or ulcerations in the throat and in the gums. Ayan. So again, this is caused by your Fusobacterium species. Yan, so lab diagnosis, so gram stain, they will appear as spindle-shaped cells with long filament with tapered ends. So ganito yung itsura nila under the microscope. So tapered ends, Fusobacterium. Ayan. So sila lang, ganito ang appearance under the microscope. So tapered ends. So, sino to? Ayan, so they also fluoresce, no, under the under Wood's lamp or under the UV light. So if we use a UV light in the agar plates, no, they will produce yellow-green fluorescence. And then in the KVLB, again, the kanamycin, vancomycin, lake blood agar, they will they have this yellow to gray colonies. So your Fusobacterium nucleatum, ayan, so varied colonial morphology. They may resemble breadcrumbs or coarse granules in your agar plates. And then colonies of your Fusobacterium nucleatum has a rancid butter odor due to the production of butyric acid. And then catalase, yan. So these are the following biochemical reactions. So negative sila sa lahat. However, your necrophorum can ferment glucose. Your nucleatum naman cannot ferment glucose. And they are indole positive. And then sa susceptibility testing, no, the potency disc test. So resistant siya to vancomycin, but it is susceptible now to kanamycin and colistin. So ganina, si Bacteroides, it is resistant to all, but your Fusobacterium is resistant lang to vancomycin, but is susceptible now to kanamycin and colistin. Ayan, so Prevotella. So next genus under your anaerobic gram-negative rats is the Prevotella. So Prevotella species are again predominant commensal organisms in the oral cavity. So this is also or these are also the agents no of a variety of oral infections. So in the agar plates, so the young colonies of your Prevotella appear tan to buff in color. However, the older colonies will produce brown pigments. And this organism is fluorescent, so they will produce brick red fluorescence under UV light. Ayan, so this one. So brick red ang kanyang fluorescence. Kanina, your Fusobacterium naman, yellow-green. Si Prevotella species can cause or can produce brick red fluorescence under UV light. So lab diagnosis of your Prevotella, we have gram-negative rods, of course, and then they are saccharolytic. So they can ferment large or many carbohydrates, indole positive, and then it is also inhibited by bile. So, difference nila is yung potency disc test. So, resistant siya to kanamycin and vancomycin but is susceptible now to colistin. Next is the Porphyromonas species. So, your Porphyromonas species, yan. So, again, they are normal floras lang. But once they go out of their natural habitat, they will now cause infections of the head, neck, oral cavity, and urogenital tract, and with pleuropulmonary infections. So, lab diagnosis of your porphyromonas. So, again, they are gram-negative rats and they are asaccharolatic. So, compared with your Prevotella species, since pareho sila mag-produce of brick red fluorescence under UV light, okay, the difference nila is that your porphyromonas, they cannot ferment carbohydrates. Okay, si Prevotella can ferment a lot of carbohydrates, but your Porphyromonas hindi. So in the potency disc test, they are susceptible to vancomycin. However, they are resistant now to kanamycin and colistin. So again, ang itsura nila under under your UV light, they produce brick red fluorescence. So how do we differentiate them? We can differentiate them by fermentation or carbohydrate fermentation tests. And also the potency disc test. So in summary, ayan, so summarize ko na for you, your Bacteroides fragilis again, they are resistant to all. Okay, kanamycin, colistin, and vancomycin. Catalase positive, indole negative, and siya lang yung mag-grow sa bile. Okay, and then Fusobacterium nucleatum naman, vancomycin resistant but is susceptible to both kanamycin and colistin. So your Prevotella species naman, they are um, resistant to your kanamycin and vancomycin but is variable or susceptible to colistin. And catalase, indole, and bile negative sila. So, si Porphyromonas naman, susceptible siya to vancomycin, but again, they are resistant to kanamycin and your colistin. Ayan. So, this is the summary now of your anaerobic gram-negative bacilli. Okay, so now we'll discuss your anaerobic gram-positive cocci. So, kumana ta with the gram-negative rods and the anaerobic gram-negative bacilli. So, isa lang man ang significant, no, under your anaerobic gram-positive cocci is your Peptostreptococcus anaerobius. So, your Peptostreptococcus anaerobius causes infections in the brain, ear, jaw, pleural cavity, and urogenital tract. So, this is the only organism that is susceptible to your sodium polyanethol sulfonate. Okay? So, di ba, mao man ni siya ang atuang additive or our anticoagulant in our blood culture. However, they are susceptible. So, paano natin siya i-counteract? So, we can counteract this action of your SPS no by addition of 1.2% gelatin. So, the addition of gelatin will counteract the action of SPS against your Peptostreptococcus. Okay, so in SPS, no, they will produce greater than or equal to 12 millimeter zone of inhibition. So your your Peptostreptococcus anaerobius naman, they are resistant to kanamycin and colistin, and indole and PYR negative and non-hemolytic sila with sweet odor on the culture media. Okay, siya lang again ang susceptible to SPS. Again, to counteract the action of your SPS, we can add 1.2% gelatin. Okay, and then next is the anaerobic gram-negative cocci. So, gram-negative cocci, again, color red sila under the microscope. Kanina, si Peptostreptococcus, that was gram-positive cocci. And then gram-negative, we have one lang, so si Veillonella. So Veillonella is a common isolate but rarely involved in the disease. So your Veillonella can also produce red fluorescence under UV light. So how do we differentiate them from the your Prevotella and your Porphyromonas? Of course, gram stain. So gram stain siya kay gram-negative cocci. So si Prevotella and your Porphyromonas, they are gram negative naman na bacilli or rods. Okay, and then Ayan, so the do's and don'ts for your anaerobic culture, again, the ideal specimen must be whole tissue or aspiration via needle and syringe. And then if you are using needle and syringe, you remove the needle following aspiration. Swabs are contraindicated. Again, they are least favorable for collection because they will be exposed to air. So we cannot isolate those anaerobic organisms. So throat swab for anaerobic culture must be rejected. And they have no special nutritional requirements. So we have vitamin K, hemine, and yeast extract, no. Kailangan nila 'yan to grow under uh anaerobic conditions. Okay, so that's it, no, for your aerobic and anaerobic gram positive and gram negative organisms. So I hope that you will study your lessons from your bacillus, no, until this topic. And then next topic will be giving you your mycobacterium na lecture video. So please study also mycobacterium species. Okay, so thank you and see you next lecture.